We have spoken about general regulation of enzymes, but now we're going to look at specific regulation of different types of allosteric regulation, both activators and inhibitors of different enzymes in glycolysis and gluconeogenesis. In this pathway, there are actually two primary sites of regulation. When we're looking at the conversion of pyruvate and phosphoenol pyruvate, and then also the conversion of fructose 1,6 bisphosphate to fructose 6-phosphate and vice versa. So you can see that inhibitors here have the lines with the kind of line at the end, and activators are actually going to have the arrow at the end. So you can see this positive arrow. That's an activator of phosphofructokinase 1, for example, this fructose 2,6 bisphosphate. I'm going to zoom in and talk a little more specifically about just one of these, that step on the top end of, the bottom end of the glycolysis pathway when we're looking at making pyruvate. Now, you can see here that if we have pyruvate, we're going back up to oxaloacetate and making phosphoenol pyruvate. Acetyl-CoA is actually a positive activator, an allosteric activator of pyruvate carboxylase. This kind of makes sense if you think about it. Acetyl-CoA, remember, is a downstream product of, this is kind of pyruvate, and then we go to acetyl-CoA, which gets transferred to acetyl-CoA in the mitochondria. And that means when we have a lot of this acetyl-CoA, that means we have a lot of product down here. And it also means that we have a lot of energy, and we have a lot of the product here. If we have a lot of product of glycolysis, that means that we might want to actually go back up this way to actually activate the synthesis of oxaloacetate, phosphoenol, pyruvate to actually undergo gluconeogenesis. So again, when we have a lot of acetyl-CoA, that activates the gluconeogenesis pathway. On the other end of things, if we have a lot of fructose 1,6 bisphosphate, that is actually a reactant upstream of glycolysis. So we're actually going to here, if we activate the pyruvate kinase, we're actually going to activate glycolysis as well. So again, if we have reactants in that glycolysis pathway, we're going to activate one pathway, versus if we have products of the glycolysis pathway, we actually might want to go and activate the reverse reaction. Now, if we look at these inhibitors, ATP and phosphorylation, let's start at ATP. If we actually zoom out back to this earlier diagram, you can see fructose 1,6 bisphosphate is up here. It's on this, right after this irreversible step. So if we have a lot of this reactant here, that can actually activate the this pathway. Actually acts as a activator of pyruvate kinase, so we can get rid of some of this reactant. So it's a good way of upstream activator of this pathway, so we can make more pyruvate. And now let's look at ATP here. So if we have ATP, it actually can act as an inhibitor of the pyruvate kinase. And that makes sense to some extent. Glycolysis, the point of glycolysis is to make ATP. And if we have a lot of it already, then that process can slow the glycolysis down, since we don't need more ATP, and potentially that can allow us to build up glucose and use that for other processes. For example, like making glycogen stores. And we'll come back to this protein kinase A in a little bit. This can also act as an inhibitor to pyruvate kinase. I just wanted to show you also some of the interactivity, the cooperativity of some of these enzymes and what they look like. And this is actually the BCC shown here in red on the left side of this image. And we're going to look at some of these enzymes. And this is actually the biotin carboxycarrier protein, or BCC. You can see it kind of fits in here on the pyruvate carboxylase here is also shown, and then there's this cooperative binding that this actually fits into. We have the CoA here, we have ATP. So if we go back to this pyruvate carboxylase, actually we can see the binding to that BCCP protein, and that's again shown here. This is kind of the three-dimensional image of that. So we can have this cooperative binding of these different subunits, and they can influence each other and change the affinity, change the shape of these enzymes, and act as either cooperative or inhibitive binding. Here's the other location of one of those irreversible steps where there's high regulation. So this is the other site of primary regulation for glycolysis and gluconeogenesis. And you'll see that one of those things that we're looking at here, both AMP and fructose 2,6-bisphosphate have opposing regulatory functions. So when there's high levels of AMP, less energetic form of adenosine, that's adenosine monophosphate instead of triphosphate. Remember, the kind of point of glucose being broken down in glycolysis is to make more ATP for our body. So if there's high levels of AMP, that means we probably have low ATP reserves. AMP is going to actually inhibit and reduce the buildup of glucose, and it's going to actually activate the breakdown of glucose. So that kind of makes sense, right? We need more ATP, less AMP, so we're going to slow down this process. We're going to break it down and hopefully make some more AMP. So we have activation of this phosphofructokinase be activated by AMP, and then the fructose 1,6-bisphosphatase, which is going backwards in gluconeogenesis, should be inhibited. The same kind of idea where the fructose 2,6-bisphosphate here is inhibiting this, and actually on this side is going to be activating this. These are both kind of ways that we can change that if this is going to be going down to glycolysis or going up to gluconeogenesis. And this regulatory mechanism kind of doubly activated here. Likewise, if there's a lot of ATP, this can also inhibit this phosphofructokinase. And citrate just kind of tells us that if we have high levels of citrate, it kind of tells us we have high energy levels. And we have lots of components for the citric acid cycle, for example, to work with, and we don't necessarily need any more. Again, some major sites of regulation here for these different enzymes. Looking just a bit closer at the protein and the protein structure here, AMP actually acts as an allosteric inhibitor, and as fructose 2,6-bisphosphate acts as a competitive inhibitor. So this is for the tight form, the inactive form of 1,6-bisphosphatase, and it's actually bound to different compounds of AMP here that's acting as an allosteric inhibitor. You can see it's trimeric, so there's 1, 2, 3, 4, actual 4 components of this. And again, this is where this is the inhibitor, and this is the enzyme, the fructose 1,6-bisphosphate that we're actually looking at over here. So the more AMP that is there, the higher the concentration, right? The more that can be bound in these different regions, and then the less active this enzyme gets. So based on this information above, what does this say about where these two factors are binding fructose 1,6-bisphosphate? So if AMP acts as an allosteric inhibitor versus fructose 2,6-bisphosphate acts as a competitive inhibitor. So are they binding in the same domain, in a different region? Remember, go back to some of the last chapters about enzymes and enzyme kinetics. Think about what that means for the active site and where, either of these you think that they bind, if it's in that active site or not. I'm not going to give you the answer right now, but if you have a question on this, come and see me in my office hours and find out. In addition to breaking glucose down into using glycolysis to pyruvate, or building it up with gluconeogenesis to create glucose molecules, I also want to briefly talk about the pentose phosphate pathway, and that's this pathway, and we're going to talk about this a little more in detail later. But two important functions of this pathway is we can take glucose and we can go through this pathway to generate our components for cofactors. So the NADH is going to be kind of made from this, as well as our DNA, our backbone for RNA and DNA. So again, we'll talk about this later, but when we have a lot of energy, when we have a lot of extra resources, we can make more DNA. We actually need a lot of resources, right, to make DNA. So if we don't have a lot of glucose, we're not going to be able to turn over and make more DNA. We're not going to do cell division. So this is really important that we have a source of glucose for making these RNA and DNA components. Now, for the rest of this discussion here, I'm going to talk about actually glycogen. Again, if we have a lot of glucose, the other thing we can do with this is to make these longer glycogen stores. So we can do the pentose phosphate pathway to generate that coenzyme NADPH and RNA and DNA. But then we can also, when we have a lot of glucose, we can store this as glycogen for quick energy reserves for our muscles. So let's just look at what that looks like. So generally glucose are these alpha 1,4 or 1,6 linkages. You can see on the first carbon, the 1, that's what we would get the 1 here, 4, and then the 6 linkage on the 6th carbon. So we can get a 1,6 or a 1,4 if it's coming off this 4th carbon here. So those are the types of linkages that we have enzymes to synthesize and actually also break down. So this is just a picture of what a glycogen compound might be. So how it's very branched. There's highly branched chains that are in specifically a lot of our skeletal muscle has a lot of glycogen stores. So this is actually an electromicrograph of a glycogen in the liver as well. Glycogen stores. Our liver, remember, has to regulate and provide blood glucose for our whole body. So there's also a lot of glycogen stored in our liver as well. So here, glycogen, glucose, we have that can turn on, get into our muscles for energy. If we have storage issues, there are certain types of glycogen storage diseases. People can have really low liver levels that can cause some problems and cause low energy output as well. So glycogen stores are really important for quick energy and for our body's ability to maintain our energy levels. So I thought this was kind of an interesting diagram that I found about what our muscles are actually using for energy. And if we look at the substrate as a percentage of oxygen consumption, so if we think about the breakdown of these compounds for respiration, most of the time, if initially when we're starting a workout, if we're doing a workout for 300 minutes, for example, which is a long workout, initially, right, in the first 20-30 minutes, most of what we're doing is burning our glycogen stores in that first half hour of working out. So once you get past that, close to an hour, you're going to start to probably have depleted most of your muscle glycogen. At that point, after about 20 minutes or so, we're going to start to increase the amount of fatty acid that we're actually using up. So here's kind of our fatty acids that we're going to be using up. We're going to start really not until the first 20 minutes or so. And so what we can see also here is our blood glucose levels kind of spike and then drop back down. But our body tries to keep these pretty regulated. And when we have most of our muscle glycogens gone initially, but then it's the energy is actually coming from our fat after about 60 minutes or so. And that's what's actually providing a majority of our energy after that. Once our glycogen, we try to regulate the glycogen stores down here, you can kind of see this kind of levels off. But once those are officially depleted after a couple of hours, that's kind of a lot of times where we say we hit the walls when our glycogen stores have been depleted completely, and then we have to completely rely on our fats for energy at that point once our glycogen stores are completely gone. We've kind of also talked about this before. The other thing to think about is if with intermittent fasting or people that do fast, they're going to probably run out of their glycogen stores a lot faster because they haven't had a chance to build them up. So some of those people might be better at, or they maybe don't feel this wall as hard because they might be more effective at burning fatty acids for energy as well. But then also they might also build glycogen stores faster when they are eating. And so that is something to think about. Our body runs more effectively when we do have glycogen stores. So it's not our glycogen stores are better energy. So if you are in a competition or doing something, you actually want to probably do some carbo loading so that you can perform more effectively, especially if you know you're not going to be doing a marathon or something where you're going to be working out for 250 minutes, for example, which most of us are not unless we're marathon runners or triathletes. So looking at this glycogenesis pathway, the first step in this pathway, if we start at the glucose 6-phosphate, so we have the 1 carbon, 2, 3, 4, 5, 6. We have the 6-phosphate, right? And we actually are going to do the phosphoglucomutase, where we're going to take the phosphate off the 6 and transfer it over to that 1. So we change it to 1-phosphate. We're not doing anything that is changing the number of phosphate groups or doing anything. We're not adding or subtracting anything. We're just moving things. So this is going to be a transferase or an isomerase type of reaction. This next step is interesting. We actually have a single phosphate, and we're going to add another one of these phosphate. We're going to actually glue this together. So we're going from this glucose 1,6-phosphate to UDP glucose. So uridine is a nucleotide, and it's got UDP diphosphate, and we're going to actually glue these things together. So this is actually going to be a synthetase. It's a ligase is the type of enzyme class. Again, this uridine is going to be from the pentose phosphate pathway. So we have a lot of energy. We should have plenty of this uridine, and that can also act to activate the buildup of glycogen stores as well. That's just a little picture of what that actually looks like, the uridine, instead of a word. So then the third step in glycogen synthesis is now we're going to take this and we're going to move this onto this growing chain, right? So we're 1,4 synthesis here. And we can see that this is going to be a ligase because we're actually going to make a longer chain again, mediated through synthase. And so it's technically it's a synthetase. So because synthases should be generally adding something to a double bond, which we're not necessarily doing, but we're adding a longer chain. But we'll give it, but this is the name of the enzyme anyway. But we're creating bigger, longer chains. So this is actually a ligase, even though it says synthase on here. Glycogenin is an enzyme, glycogen synthase. This is the primer. So we actually have to have, before we can kind of really go to town on this, we have to have 8 glucose residues that kind of start this seeding process. There's actually a protein in the middle of this, and the glycogenin adds 8 glucose molecules to itself. So this is the enzyme, this is the protein glycogenin in the middle here, and it's going to kind of add those glucose molecules to itself to seed itself, and then the polymerization, the glycogen synthase can then start adding onto that chain after that seeding has been done. So usually that that binding is going to happen after that has been seeded. So there is a glycogen storage disease I should mention as well. They found defective in glycogen storage and they have problems with this enzyme actually functioning normally. This results in muscle cells with the inability to store glucose and a lot of times weakness in muscle and energy, low energy levels, and sometimes heart disease as well. So we've built this big chain or these big groups of glycogen. And now glycogenolysis is actually when we want to do the reverse reaction and start breaking down these glucose molecules to use in glycolysis, for example. When we have these long chains, we actually go the reverse reaction. We're going to have inorganic phosphate shown here. Glycogen phosphorylase is going to transfer this, and it's not a hydrolysis necessarily. It's a transferase because we're actually not adding water to this molecule. We're actually adding inorganic phosphate, and we're going to break this bond. We're going to cleave that bond. So this is going to break into this longer chain of glycogen and then a freed chain of glucose 1-phosphate. So we have this freed glucose molecule. The problem is it's glucose 1-phosphate, not glucose 6-phosphate, which we know is the starting point. So we can actually again use the phosphoglucomutase. That's going to convert whatever the highest levels are in the cell. So if we have the most alpha D glucose 1-phosphate, it's going to convert it to glucose 6-phosphate. If it's vice versa, we'll have a lot of glucose 6-phosphate. We'll transfer it to glucose 1-phosphate to get to our equilibrium state there. So wherever we have high levels, that'll help convert and change over to wherever we need it in our cell the most. Kind of think of I don't know if anyone's ever seen this, probably not. This is a really old show, The Fraggles. Just kind of reminds me of switching from breaking down to building up. These guys, these little dozers, are always building up these things, and then these little Fraggle guys are eating them all the time. So they're constantly building, and there's constantly build up and break down. Kind of think of that for the glycogen stores as well. But what's really telling our cells if we need to start building up or breaking down? So we're going to talk about signaling a little bit here, hormonal signaling. There's a couple different signals we'll talk about and introduce you to insulin, which I'm sure you've heard of already. There are insulin receptors that are going to bind to that hormonal signal. And what that actually does in our cell is it actually recruits the insulin binds, and once it's bound, it actually activates the recruitment of these vesicles that bring in our GLUT4. So these are glucose transporters that we were talking about before. So remember the glucose that's floating around and it gets transported. It's active transport, right? It's active transport through those transporters. So that's why it's important. Glucose doesn't get absorbed by the cell. It actually gets actively transported. Insulin brings those transporters to the membrane surface so that glucose can be taken up. If we stop responding to insulin, we stop bringing those transporters, and then our cell doesn't get enough glucose in it because we're not transporting glucose anymore. Another hormonal control mechanism is through glucagon, and this glucagon binds a receptor. It's a little bit different of a hormonal signal. So this actually activates a signal transduction mechanism. So glucagon binds to that receptor, and then that activates an adenylyl cyclase. So this enzyme actually converts ATP to cyclic AMP. So this is actually cyclic AMP, where we have it's cyclic because we make this circular shape here. Instead of having the phosphate group come off the end, it actually kind of creates this phosphobond on that same sugar molecule here at the bottom. So cyclic AMP is actually a signaling molecule that can actually activate then this PKA. Protein kinase A becomes activated, and then this is kind of attached to that PKA and its active enzyme. We talked about this before, and it can regulate different components of the glycolysis pathway as well. this is the big long kind of chain of what it can do. So active pKa can then regulate the process here of phosphorylase kinase, which can then regulate glycogen phosphorylase, which can regulate this glycogen synthase. So all of these things actually regulate glycogen as well as glycolysis as well. So the breakdown of glycogen, this kind of makes sense if we have glucagon. These levels of glucagon are released when we have low glucose levels. So if you follow this pathway, you can see that we have with low glucose levels, we activate this pathway. And so this active pathway is going to, through multiple different mechanisms, slow down glycogen synthesis. So that makes sense. We don't have glucose. We shouldn't be building up our stores of glycogen. We need to be using up our stores of glycogen. So that this pathway kind of makes sense there. This is an interesting experiment that was done a while ago where a man consumed 100 grams of glucose and then he fasted for 40 days. And this is what happened when he did that. So the exogenous levels of glucose he started with were labeled with a dye. So we're actually that in the red here, you can see those levels of exogenous glucose were labeled in his bloodstream with a dye so that the doctors that he was working with could monitor that. You can see that those levels were essentially consumed after 4 hours of fasting. So not that long for 100 grams of glucose to be consumed. Then what happens next is we actually have in the blue here, after about 3 or so hours, we have those glycogen stores being used up. And those glycogen stores are continuing to be used up until about 28, 30 hours, where the glycogen stores are completely used. So now he's not exercising. The last kind of picture I showed you with the oxygen consumption, that was someone that was working out for a number of hours. But this person's theoretically not working out. He's just kind of surviving. So the glycogen stores can last for over a day for the 24 hours. What you can see here are, besides the exogenous glucose and then the glucose from the glycogen stores here, we can actually also now, they're kind of showing us the glycogen stores from gluconeogenesis. So once we start building up, when we start using the buildup of the glucose levels from the creation of glucose from gluconeogenesis, so liver-derived glycogen. And you can see that allows us to kind of regulate our glycogen levels. If we look at total amounts of glucose here, right, this kind of stays regulated because we're going to build up, not only are we using like glycogen stores, but we're actually building up new glucose from gluconeogenesis. And we can kind of regulate that out to about, and maintain that for about 2 days here. At about 2 days, then we kind of slow down our metabolism, you can see, and we reduce the amount of blood glucose that we have, and that can be regulated out to about 40 days. So we start to transfer over some of the stuff we can do. So this is, so we can see the absorptive phase where we're taking up that glucose and our cells are taking it up, our liver is taking it up, and we're maybe building our glycogen stores. Then we start, after that's gone, then we start to use our glycogen stores up, and then we then start to create and turn on. Right about this point, right, we start to turn on our other pathways for breaking down glycogen and building up glucose from gluconeogenesis. So this would say that probably right about here is where we have a transition from where we have our insulin levels are high and then glucagon levels are high from here on out, because we're trying to now build up the gluconeogenesis, break down the glycogen stores. So that's going to help activate those processes. And the insulin is going to help us absorb some of that glucose that we have in our bloodstream. Just as we talked about different ways of regulating glycolysis and gluconeogenesis, there also we can regulate glycogen metabolism via glycogen phosphorylase. So this is glycogen phosphorylase. It's a dimer here, you can see just kind of a cartoon version on the left, and then more of a three-dimensional protein structure on the right. There's an actual glycogen binding site and the catalytic site in here. So it's a partially processive. Unlike glycogen synthase, it kind of quickly activates that breakdown of phosphorylation and breaking down of glucose. There's a catalytic site here, and then we can also have glycogen that binds here as well. I wanted to look at glycogen phosphorylase regulation a little more closely. I want to tell you that there is a difference between what's happening in the liver versus the skeletal muscle, but we're going to look at skeletal muscle form here right now. And there are two states that you can kind of see here. There's a relaxed state or an R state and a tense state or a T state. And this is the active site circled in red, and that's where we have a dimer, two different enzymes coming together. They work in a pair. Because the relaxed state, the enzyme active site is more open and available, it works better. It is quicker and more functional. The tense state, this is kind of somewhat blocked in the enzyme, and it doesn't work as effectively. A few things regulate these different states. One is the phosphorylation event. So there is a kinase and a phosphatase. That kinase would add the phosphate groups, or a phosphorylase would take them off. And these are going to somewhat change the phosphate groups, phosphorylation groups. When we are in the absence of glucose, this is the state that is formed. But if glucose is present, then this phosphorylated form goes down into this tense state, and it's less active. So this is the active form of glycogen phosphorylase. And we also have the relaxed state when we are unphosphorylated in this instance, and we are in the presence of AMP. So this means that AMP, monophosphate, means that we have less energy. We have a lot of this energy has been used in the presence of AMP, adenosine monophosphate. That means that we don't have the triphosphate form, which is higher energy. This is going to be the state that is preferred. In the presence of glucose 6-phosphate and ATP, which means we're in the fed state, that is going to allosterically regulate this and form this tense state, which is going to reduce the amount of glycogen available in our system, or glucose, available, which makes sense, right? If we have a lot of energy and already glucose 6-phosphate available, we don't want to break off other glucose. We don't want to break down glycogen to form more glucose units. So these are, depending on if they're phosphorylated or not, regulated by either glucose or glucose 6-phosphate, ATP, and AMP. Now, you can kind of see what's happening with the regulation when we have the activity of glycogen phosphorylase in the presence of ATP or glucose 6-phosphate. So we normally have, this is the uninterrupted enzyme activity. The Km is a lower number. And remember, the Km is a relative readout of how well it binds at 50% capacity. So the lower the concentration number, it's saying it can grab things better than, for example, when the presence of ATP or glucose 6-phosphate, it reduces that activity or the ability to bind. So that affinity is reduced because this number is bigger. We need to have more of a concentration of substrate in order for half of the sites to be bound. So this slows down the speed of the enzymatic reaction. Now we can also look at when we have adenosine monophosphate present, and that is going to reduce the Km number, which means that we're increasing the affinity. So again, the smaller this number means that this at half max, that means half of those enzyme sites are active and bonded to the substrate. And that lower that number, the better that enzyme is able to grab stuff at lower concentrations. So this is what I wanted to discuss in terms of the regulation, and
we'll continue looking at some of the other enzymatic processes and metabolic processes in later lectures.